Welcome to this session on computational biology. Today, we are going to explore a fundamental statistical tool known as the z-test. In the world of biology, we constantly observe changes and differences, but we must always ask ourselves, are these differences real or just random noise? By the end of this video, you will understand exactly how the z-test answers that question, allowing us to make confident, mathematically backed claims. Let us start with a simple, intuitive scenario. Imagine you are a baker who makes loaves of bread that usually weigh exactly 500 grams. One day, you buy a new type of flour, and you want to know if it changed the average weight of your bread. You bake a batch of 50 loaves and find their average weight is now 510 grams. This brings up a crucial question. Is this 10-gram difference just random luck, or did the new flour actually change the weight? This is the exact type of problem we face in biology every day. Now that we see the problem, how do we solve it? This is where our mathematical detective steps in. The z-test is designed to help you answer this exact question. It tells you whether a new group of things is genuinely different from what you normally expect, or if the difference is just a random fluke. Instead of guessing, the z-test gives us a rigorous way to measure confidence, ensuring that when we see a change, we know whether to trust it or ignore it. Building on that intuition, let us look at the formal definition. In statistics and computational biology, a z-test is a specific type of hypothesis test. We use it to determine whether the mean of a sample is significantly different from the known mean of a population. The test calculates what we call a z-score. This score measures exactly how many standard deviations your sample mean is away from the population mean. By looking at this z-score, we can find the probability that the observed difference happened purely by chance. So, when exactly can we use this test? The z-test is specifically applied under two main conditions. First, the sample size must be large, typically when n is greater than 30. Second, the standard deviation of the entire population must already be known. When these conditions are met, the test is highly accurate. If the test shows that the probability of the difference happening by chance is extremely low, we reject the idea of random chance. We then confidently conclude that there is a statistically significant difference. Why do we care about this specifically in computational biology? Imagine you are testing a new drug designed to lower blood pressure, and you know the average blood pressure of untreated patients. When you give the drug to a large group, their average blood pressure drops. Without the z-test, you would just be guessing if the drug worked, or if those specific patients just happened to have lower blood pressure that day. The z-test provides a rigorous mathematical proof that your biological findings are real and reliable. It is the absolute foundation for making confident claims in genetics, epidemiology, and clinical trials. Now that we understand the concept, let us look at the mathematical representation. The formula calculates the z-score by taking the raw difference between our sample and the population and dividing it by the standard error. Let us define each symbol. Z is the z-score, our final standardized value. X bar represents the mean of our specific sample. Mu represents the known average of the entire population. Sigma is the population standard deviation, which tells us how spread out the data normally is. Finally, n is the sample size. Notice the denominator. If the sample size n increases, the standard error shrinks, making the resulting z-score larger and more statistically significant. Let us apply this formula with a concrete example. Suppose a biologist knows a plant species normally grows to an average height of 150 centimeters, with a population standard deviation of 20 centimeters. A new fertilizer is tested on a sample of 100 plants, yielding an average height of 155 centimeters. We want to know if the fertilizer actually worked. We plug our known values into the formula. The numerator becomes 5, representing the raw difference in height. For the denominator, the square root of 100 is 10, and 20 divided by 10 gives a standard error of 2. Finally, dividing 5 by 2 gives us a final z-score of 2.5. So we have calculated a z-score of 2.5. What does this number actually mean? In statistics, a z-score greater than 1.96 corresponds to a standard 95% confidence level. Because our score of 2.5 is strictly greater than this critical threshold, it falls into the rejection region. This means we can confidently conclude that the new fertilizer significantly increases plant height. The result is not just random luck. The biologist has successfully created an effective product, and the math proves it. Let us look at another critical example in computational biology. Researchers are studying a gene that produces an average of 300 RNA transcripts in healthy individuals with a standard deviation of 40. A sample of 64 cells from a diseased patient shows an average transcript level of 285. Is this drop significant? We plug our values into the formula. The numerator evaluates to -15, showing the sample mean is lower than the population mean. The square root of 64 is 8, so our standard error is 40 divided by 8, which is 5. Dividing -15 by 5 yields a final z-score of -3.0. Now that we have our z-score of -3.0, let us interpret it. If the resulting z-score is less than -1.96, it falls into the lower rejection region. Since -3.0 is much further from 0 than our critical boundary, we conclude that the gene expression in the diseased cells is significantly reduced. This is not a normal fluctuation. This gives the researchers strong mathematical evidence that the disease negatively affects this specific gene, allowing them to proceed with further biological experiments with high confidence. To truly understand what these numbers mean, let us visualize the standard normal distribution. The bell curve you see represents all the possible sample means we might get just by random chance. The large blue area in the middle is the 95% confidence interval. If our z-score lands here, the difference is just random noise. However, the red areas on the far left and right are the rejection regions. Notice where our two examples landed. The fertilizer example gave a z-score of 2.5, landing deep in the right rejection region. The gene expression example gave -3.0, landing in the left rejection region. Both results are statistically significant. As we wrap up, let us review the key takeaways. The z-test is an incredibly powerful statistical tool that helps us separate true biological discoveries from random noise. By comparing a sample mean to a population mean and factoring in the sample size and natural variation, it provides a standardized z-score. This score tells us exactly how unusual our sample is compared to normal expectations. Whether you are testing fertilizers, drugs, or gene expression, mastering the z-test allows you to make confident, mathematically backed claims in computational biology. You now have the foundation to apply this to real-world data.